Hello students, we are going to see about LC-MS. In this session, we will now study the analyzers, that is the mass analyzers which are used. So the typically mass analyzers, or you can say mass spectrometers which are used in LC-MS are triple quadrupole. You can write it as triple Q or TQMS, and quadrupole time of flight, that is QTOF. So these are the most commonly used tandem mass spectrometers. Let us first see about the quadrupole analyzer. So it consists of three quadrupoles in a row, Q1, Q2, Q3. As you can see in the diagram, three quadrupoles are used. So here Q1 and Q3, they will act as mass filter, whereas Q2, it acts as a collision cell. In Q1, the precursor ion is selected and afterwards it is dissociated into the product ions in Q2, that is in the collision cell. This process is followed by a mass scan in Q3. So again the ions will be selected in Q3, that is third quadrupole. In the collision cell, collision-induced dissociation of the precursor ion takes place. Energy is transformed through the collision with neutral molecules, that is collision gases like argon, helium or nitrogen are used, resulting in bond cleavages and rearrangements of the selected ion. Here you can see the diagrammatic representation. Here you can see the schematic diagram. Here first selection takes place, then collision with help of the gases like nitrogen, argon, helium. Again the second selection and finally the detection. So as here three quadrupoles are used, it is known as triple quadrupole. In next it is quadrupole time of flight analyzer. It has a quadrupole analyzer and a time of flight analyzer separated by a collision cell. So in earlier, two quadrupoles were separated by the collision cell, whereas here a quadrupole and TOF analyzer, it is separated by a collision cell. The quadrupole can be used either to transmit the ions or to isolate a specific precursor ion which is then fragmented in the collision cell. So a small fraction of ions are first pulsed into the TOF analyzer by a modulator and subsequently accelerated into high vacuum field free region by applying high voltage. Ions with different m by z values travel at different velocities in the flight tube and are separated from one another. So you can say principle is same as that of the TOF, which is used in mass spectrometry. TOF analyzers, they offer high mass resolutions while being able to scan over large mass ranges quickly. Okay, now having studied about the ion sources or you can say interfaces and analyzer, now let us see about the advantages of LC-MS. So first is specificity. So selection of precursor ion with the first quadrupole and product fragment into the second quadrupole by tandem mass spectrometer, it exhibits high specific detection. Again, this technique allows for a good flexibility. So new assays can typically be developed with a high degree of flexibility within a short time. Abundance of information. A single analytical LC-MS run due to fast ion selection electronics, multi-parametric, then quasi-parallel analysis, it can produce a large number of quantitative or qualitative results. Then sample preparation here is simple and does not require derivatization technique. LC-MS, they are generally optimized to shorter run times. Therefore, far higher sample throughput can be obtained when compared to GC-MS. Now let us see the applications of this technique. So first application is drug metabolism and toxicology study. Now as we know, whenever a drug enters in our body, it undergoes metabolism, and this metabolic pathway, they are divided into phase 1 and phase 2 reactions. Okay, so the information required to determine the metabolic fate of a NCE, that is new chemical entity, it includes detection of metabolites, structure characterization and quantitative analysis. Okay, so this information about the new drug molecule is very important, that what is the fate of that drug molecule once it enters the body, because it can get converted to sometimes toxic metabolite also. So the characterization and quantification of these metabolites is necessary. So it is possible with help of LC-MS. It offers superior specificity, sensitivity and efficiency. Next application is quantification method for biogenic amines. Now, quantification of biogenic amines is mainly used for diagnosis of neuroendocrine tumors. Now, many of the hormones, or you can say neurotransmitters in our body, they are chemically the amines. So, in the conditions like pheochromocytoma and carcinoids, the quantification of these biogenic amines is important. So, this can be achieved with help of LC-MS. It greatly increases the sensitivity as well as specificity of the test results. Next is the newborn screening. Okay, so the initial foray of LC-MS into the clinical arena was its application to newborn screening of the inborn errors of metabolism. It is used in routine newborn screening for inborn errors of metabolism. At present, newborn screening in dried blood spot encompasses more than 30 diseases. That is, by analyzing the dried blood spots of the newborn, we can analyze 30 diseases, which include organic acidemias, then amino acidopathies, then defect of fatty acid oxidation. Now, this defect, fatty acid oxidation, it is related to sudden infant death syndrome. Okay, so all these diseases, they can be found out by analyzing the blood spots, which is possible with help of LC-MS. Then it is used for therapeutic drug monitoring for immunosuppressants. Now, immunosuppressants, they are generally used in case of autoimmune disorders or wherever there is organ transplant, like liver transplant, kidney transplant. So at that time, to reduce the body's immune responses, immunosuppressants are used. Now, the therapeutic window of this immunosuppressants is very important, as they have narrow therapeutic ranges. So, its therapeutic monitoring can be done with help of LC-MS. It provides high specificity and sensitivity for immunosuppressants. Next application is in the field of endocrinology and steroid testing. Radioimmunoassay and enzyme immunoassays, they were the main techniques for measurement of steroids in clinical laboratories. But they have limitations like lack of specificity, then limited dynamic range and matrix effect. So nowadays, LC-MS is viewed as an innovative analytical technology applicable to wide number of analysis in the endocrinology laboratory, mainly the quantification of various steroids. So analysis of 25-hydroxyvitamin D3 and D2 levels by LC-MS has gained wide momentum. LC-MS methods, they have been developed for a variety of steroids like testosterone, aldosterone, cortisol, progesterone, estriol, etc. So you can see these are the various steroid hormones found in the male and female bodies. Next application is drug of abuse and pain management testing. So for this purpose, GCMS is still most widely used, but LCMS is gaining ground in that area due to simple sample preparation, as we have discussed in advantages. Here there is no requirement for derivatization and shorter run times. Direct drug screening from the urine has been reported, and methods are available for multi-drug panels for drug of abuse screening. Also, methods are there for confirmation of benzodiazepines, then measurement of drugs like buprenorphine and its metabolites, amphetamines, opioids. So you can see these are the habit forming drugs which are often abused. So its analysis can be done by LCMS-MS. Therefore, LCMS-MS finds application in drug of abuse and pain management testing. Okay, now let us see the limitations of LCMS-MS. So first limitation is matrix effect. Due to matrix effect, ion separation or enhancement can occur, which is caused by the sample matrix and interferences from the metabolites, which leads to decrease in selectivity. Matrix effect occur when molecules coeluting with the compound of interest alters the ionization efficiency of the electrospray interface. This matrix effect originates from the competition between the analyte and the coeluting undetected matrix component. These are the result of competition between non-volatile matrix component and analyte ions for access to the droplet surface for transfer to the gas phase. Matrix effects are also compound dependent, and chemical nature of compound is a significant effect on the degree of matrix effect. So how to overcome this matrix effect? Sample preparation techniques. So this can be reduced by injecting smaller volume, or you can dilute the sample. However, it will hamper the sensitivity. So proper sample cleanup is of prime importance to reduce this matrix effect. The next is chromatographic separation. Improved chromatography is a simple way to separate interfering compounds from analytes. So here you can use gradient elution, which can help to wash the column after injection and prevent late eluting compounds from the previous injection to the interface. So the late eluting compounds from the previous injection to interfere, so that they would not interfere. Then use of internal standard. Here you can make use of an isotopically labeled internal standard which experience similar ionization, separation or enhancement, and thus they can be used to compensate for the matrix effect. Next limitation is carryover. Analyte peaks may appear in the blank injection run after a high concentration sample as a result of sample carryover. So to remove this, cleaning protocols it should be used, such as repeated blank injection. Then you can do needle washes and column conditioning to ensure sensitivity of the analysis is maintained. Next limitation of LCMS-MS is sample loss. So analyte like protein, DNA, they may be lost due to non-specific binding to the laboratory consumables, such as inner surface of microcentrifuge tube. Absorption of analyte impacts the accuracy and precision of the assay. Then analyte loss can be minimized by using the containers that have low surface adhesion. Okay, so this will prevent your sample loss. Another approach is to add blocking agents that minimize the interaction of analyte with the inner surfaces of the containers. Next limitation is mobile phase buffer selection. Now, as the column eluent has to be removed prior to MS analysis, only volatile buffers can be used, like ammonium formate or ammonium acetate, which will not precipitate in the source. So here again, our choice of buffer, it gets restricted because we can use only the volatile buffers. Next limitation is cost. It is expensive and it requires high maintenance, and maintenance costs are also high. Then technical expertise, that is, here obviously good technical expertise is required, because here we are going to handle liquid chromatography as well as the tandem mass spectrometer. So good expertise is required in this field. Okay, so this was all about the LC-MS/MS, the analyzers used in LC-MS/MS, and its advantages, limitations and applications. Thank you.